What is signal transduction? Any cellular process that converts one kind of signal or stimulus into another. Most signal transduction processes involve ordered sequences of biochemical reactions inside a cell that normally leads to a cellular response to the initial signal. Note: signal does not have to cross the plasma membrane, and does not have to involve a molecule. What are the four properties of signal transduction pathways? 1. Specificity to signals. 2. Amplification of signals. 3. Desensitization, adaptation to constant signals. 4. Integration of multiple signals. Signal SADI. How is specificity important to signal transduction? Receptors specific and sensitive to type of stimulus received, typically binding to very specific molecular signals called ligands. Signals must fit, and be the only molecule to fit, binding site on receptor. What three factors account for the extraordinary sensitivity of signal transducers? 1. High affinity of receptor to types of signal received. 2. Positive cooperativity in receptor binding to ligand. 3. Amplification of signal as it is transduced down a signal cascade. How is amplification important to signal transduction? When enzymes activate enzymes, the number of affected molecules increases geometrically in an enzyme cascade. Does a constant signal contain information? Explain. No. Information theory. Signal must indicate a change in order to give information, i.e., something to track, subtract from, or compare to. Otherwise no way to use signal to modify behavior or response, since the behavior already has constant modification built in as a natural adaptation. What are two reasons for desensitization and adaptation mechanisms to exist? 1. To shut down signal pathway if signal is constantly there. 2. To modulate or bring detection within a range where signal is present. Do integrated signals need to have opposite effects to be integrated? No, can have two positive or two negative signals. What is another example of integration in addition to the integration of two direct signals? Many antagonistic signals in separate signal transduction pathways often modify behavior of others pathway and an integration of response rather than integration of direct signal itself. What is the common biochemical result of many, maybe most, signal transduction pathways? Phosphorylation and dephosphorylation, though possibly more of a post-translational modification event. What are the 6 basic types of signal transducers in cells? 1. G-protein-coupled receptors. 2. Receptor tyrosine kinase. 3. Receptor guanylyl cyclase. 4. Adhesion receptor. 5. Gated ion channel. 6. Steroid receptor. Good receptors regularly adhere to. Groups of 6. What are G-protein-coupled receptors? Involve G-protein or GTP binding at the top of its enzymatic cascade. What are receptor tyrosine kinases? Directly phosphorylate tyrosines to start a kinase protein enzymatic cascade. What are receptor guanylyl cyclases? Converts GTP to cyclic GMP to start its enzymatic cascade. What are gated ion channels? Open or closed depending on ligand binding or change in membrane potential. What are adhesion receptors? Bind part of extracellular matrix and transduce this signal to cell cytoskeleton. What are steroid receptors? Bind steroid hormones that diffuse across the membrane. The 6 types of signal transducers differ in their initial step of transduction. How are they similar? Most of resulting signal transduction systems are built using 10 general types of protein components, which are reused for many different signal transduction purposes. Why are 6 types of signal transducers not considered signal transducer receptors? Voltage-gated ion channels in particular are not receptors, don't bind specific molecules to cause transduction or conversion of a signal. What is a scattered analysis? Describe the associated equations and graph. Determines the dissociation constant for a ligand-receptor complex. How is a scattered analysis done? 1. Determine amount of bound ligand at given total ligand concentration, historically by radiolabeling. What is typically used today instead of radiolabeling? Anything seen by a spectroscopic method, fluorescent spectroscopy or UV vis spectroscopy. How is the scattered analysis complicated? How is this remedied? Nonspecific binding ligand to cell membrane and other proteins, subtract out by adding large amounts of unlabeled ligand, which competes with specific bindings, but not nonspecific binding. Why does excess unlabeled ligand added to a scattered analysis not compete with the nonspecific ligand binding? Much harder to saturate nonspecific bindings. Kd of nonspecific binding is much higher than specific bindings. Huge number of nonspecific sites. How can the scattered analysis be used to determine Kd and Bmax? Describe associated equations and graph. Rearrange equations to plot line -1, k equals slope, b max equals x-intercept. When does the assumption of linearity for the scattered analysis break down? Any allosteric reactions affect linearity. Describe the structure and location of the G-protein coupled receptor. Integral membrane proteins comprised of 7 transmembrane helices. Why are GPCRs currently the most important class of proteins in human health? %70 of drugs on market target 1,000 GPCRs predicted to exist in human genome. Describe the 7 steps of the GPCR signal transduction event and signal cascade. (1) Ligand binding, e.g., epinephrine. 2. GPCR causes GTP protein to replace bound GDP with GTP. 3. Alpha subunit is released from trimeric G protein and associates with membrane-bound enzyme adenylyl cyclase. 4. Adenylyl cyclase converts ATP to CAMP. 5. CAMP is second messenger that activates pKa. 6. pKa affects cellular response by phosphorylating other cellular proteins. 7. CAMP ultimately converted to AMP by PDEs or cyclic nucleotide phosphoryldehydrases. Describe the activity of the alpha subunit of GDP during GPCR activation. Activated alpha subunit eventually hydrolyzes bound GTP to GDP, becoming deactivated, then reassociates with beta and gamma subunits. How can a beta-adrenergic GPCR be identified? Epinephrine is the ligand. What 2 main sets of factors regulate G proteins? 1. GTP, GDP exchange factors, GEFs. 2. GTPase activator proteins, GAPs. What is the function of GEFs? What is an example of a GEF? Exchange GDP for GTP to activate G proteins. GPCRs themselves are GEFs. What is the function of GAPs? aka regulators of G protein signaling, or RGSs, contribute critical Arg9 to GTPase active site of G proteins, increasing activity by 100,000-fold, bringing G protein full cirralcy. How does desensitization occur in G proteins when a signal persists? (1) Beta-gamma subunit recruits beta-gamma adrenergic receptor kinase, beta-ARC, to receptor. 2. Beta-ARC phosphorylates several serines near C-terminus of receptor. 3. Phosphorylation activates beta-arrestin, beta-ARR, bind receptor, preventing G protein from interacting with receptor. 4. Beta-ARR triggers endocytosis of receptor, which sequesters it from cell surface. Desensitization complete. 5. Eventually beta-ARR dissociates and G protein receptor is dephosphorylated and returned to cell. What attenuates the response to epinephrine in addition to desensitization? PDEs that converts CAMP to AMP, removing that second messenger. Describe the 9-step epinephrine cascade in hepatocytes leading to release of glucose into bloodstream. What is a second messenger? A diffusible signaling molecule that is rapidly created or secreted in order to transduce a signal via binding to effector proteins. What is the prototypical protein kinase in the hepatocyte epinephrine cascade that starts phosphorylation cascade? pKa, observed in many signal transduction pathways. How is pKa activated? 4 cyclic AMP molecules bind to the 2 regulatory subunits on the dimeric enzyme, pulling the inhibitory sequence from the substrate binding cleft and freeing the enzyme to phosphorylate serine or threonine on a target protein. What does pKa target? Give an example of an enzyme with this type of target. Specific proteins that contain consensus sequence XR, RK, X, Saint B, phosphorylase B kinase has 2 kinase subunits each with their own consensus sequence specificities, alpha, BEFRRLSI, beta, RTKRSGSV. Why doesn't glycogen phosphorylase have a consensus sequence? B. C. It doesn't target phosphorylate, a protein, rather it degrades glycogen by releasing glycogen 1-phosphate from the terminal alpha-1, 4-glycosidic bond. List some signals that use CAMP as a second messenger. Corticotropin, dopamine, epinephrine, follicle-stimulating hormone FSH, histamine, luteinizing hormone LH, thyroid stimulation. What are 4 factors that allow CAMP to be used in so many signal transduction events? One, many signal-specific GPCRs exist. 2, different GPCRs activate Gs or GI. 3, different cell types express different GPCRs, G proteins, and target for pKa. 4, localization of signal cascade machinery. W, in cell. What does the existence of multiple GPCRs and G proteins allow? Signal integration. Describe a common signal cascade machinery using AKAP. A kinase anchoring protein, AKAP multivalent adapter proteins, bind R subunits of pKa and localize pKa and other signaling molecules, e.g. phosphatases and phosphodiesterase, to discrete locations, e.g. to adenylyl cyclase in plasma membrane. Describe the regulatory process after a ligand binds a GPCR and the alpha subunit binds to phospholipase C in the membrane. Activated phospholipase C cleaves pep2-IP3 and diacylglycerol, both potent second messengers that work in concert. IP3 binds circuit2 channels in her, releasing circuit2 into cytosol, circuit2 and diacylglycerol activate protein kinase C and start phosphorylation cascade. What establishes the concentration gradient of calcium ions across the air membrane? SCRCA pumps. How do circuit2 ions activate other kinases in addition to protein kinase C? Through binding to calmodulin. Circuit2-bound calmodulin binds calmodulin-dependent protein kinases, which start other phosphorylation events. How does calmodulin activate protein kinases? After calmodulin binds 4 circuit2 ions, its 2 domains wrap around a specific helix of kinase. As the regulatory subunit of phosphorylase B kinase, SHAT does calmodulin allows circuit2 ion-induced muscle contraction to stimulate glycogen breakdown. How are localized sharp rises in circuit2 created? Low, medium, and high concentrations of IP3 create blips, puffs, and waves of circuit2. What is the relationship between localized circuit2 concentrations and localized CAMP signaling? Machinery concentrations. Changes in the level of CAMP can feed back to influence the level of circuit2 by acting on both circuit2 channels and pumps. In cardiac and skeletal muscle, the activity of one type of circuit2 channel is enhanced by CAMP. Describe the receptor tyrosine kinase signal receptor transducers. Intrinsic protein kinase activity phosphorylates tyrosines on target proteins, including itself. Single subunit protein ligand binding domain can vary quite a bit to accommodate a wide variety of ligands. What is one exception to the RTK having only a single subunit? Insulin receptor with 4 subunits. Describe the binding site of the insulin receptor. Prototypical protein kinase domain uses activation loop (BLU) to block substrate binding site. When ligand binding domain binds insulin, tyrosine kinase's active site allows phosphorylation of 3 tyrosines in activation loop, causing loop to swing out and allowing active site to bind and phosphorylate target proteins. Describe the regulation of gene expression by insulin through a MAP kinase cascade. What does MAPK stand for? Mitogen-activated protein kinase. Which enzyme in insulin gene expression is a MAP kinase? ERK, since it phosphorylates transcription factors that stimulate genes related to mitosis. Since MEK is an MAPKK, what does that naturally make RAF1? MAPKKK, so the insulin regulation of gene expression cascade from RAF1 on down is known as a MAPK cascade. What is the relationship between insulin and epinephrine signaling pathways? Insulin signaling usually trumps epinephrine signaling. Describe the crosstalk between the insulin receptor and the beta-adrenergic receptor, or other GPCR, that ends with GPCR internalization. Insulin binds to its receptor and receptor phosphorylates the beta-adrenergic receptor directly onto tyrosines and indirectly through PKB onto serines, which triggers internalization of the beta-adrenergic receptor. In addition to the insulin signaling pathway, what other process involves the internalization of beta-adrenergic receptors? Desensitization of epinephrine pathway due to constant presence of epinephrine. Describe the crosstalk between the insulin receptor and the beta-adrenergic receptor, or other GPCR, that ends with altered gene expression. One, insulin binds to its receptor. 2. Receptor phosphorylates the beta-adrenergic receptor directly on 1. Tyrosine. 3. Phosphorylated TYR attracts SHUSH2 containing proteins like GRB2 to bind. 4. Insulin-like signaling cascade that ends with altered gene expression. What are the implications of crosstalk? Reflects level of complexity and integration, which makes study difficult. Describe the function of guanylyl cyclase receptors. Bind different ligands and convert GTP to the second messenger cyclic GMP. How does cyclic GMP mediate its actions? Via the activation of cGMP-dependent protein kinases (PKG), which phosphorylates serine and threonine residues on target proteins. What are two physiological functions that use guanylyl cyclases? What level of functions are these? One, ion transport and water retention in the kidneys and intestines. 2. Relaxation of cardiac and smooth muscle. 1. Organ. 2. Tissue. What is the ligand nitric oxide involved in when it binds to a guanylyl cyclase? What is special about the ligand? No. Cardiac muscle relaxation, ALB, to diffuse to cell membrane to bind its receptor inside the cell. What type of receptor binds no and how does it differ from most others? Receptor includes heme group that binds no and is not an integral membrane protein and is soluble unlike most other receptors. What early drug targets the no receptor? What disease does this drug treat? What is special about the drug's delivery? Nitroglycerin, angina pectoris, pain in the heart due to contracting too hard, heart is pumping well deprived of all needed oxygen, taken sublingually, absorbed into bloodstream under tongue. Deactivation of drug would occur in liver if absorbed intestinally. What is significant about the multivalent nature of many signaling proteins? Allows interaction with multiple proteins and/or signaling molecules. What are some common binding domains of multivalent signaling proteins? What are the functional benefits for multivalent proteins to serve as scaffolds for multiprotein complexes? What level of function are these benefits? Localization of part or all of the signal pathway within the cell. Creation of local concentration effects allows reuse of second messengers, can speed up signal transduction through a pathway, serves as a point of regulation for signaling pathway, mostly biochemical, some chemical with localization. How are protein interactions of adapter proteins modulated? Give an example. Phosphorylation, SHUSH2 domain of the SRC protein binds specific phosphorylated tyrosine on a substrate protein, allowing SRC to phosphorylate other tyrosines. Phosphorylation of SRC protein itself inhibits its own ability to bind and phosphorylate substrate proteins. In addition to SRC, give another example of the regulatory mechanism of autoinhibition. GSK3 is used in another part of the insulin receptor signaling pathway. What other molecular entities provide localization, organizational, and regulatory benefits like the multivalent adapter proteins? What is one possible result of receptor activation here? Membranes, specifically membrane rafts, specific receptors and their complementary signaling. Proteins are localized to membrane raft. In some cases, activation of receptor are downstream. Signaling protein allows release of some entity from membrane raft. What are gated ion channels? Major class of signal transducers that convey signals by changing concentration gradients and electric potentials across membranes. What drives the flow of ions when an ion channel opens? What is the flow potential for a typical neuron? Concentration gradient and membrane potential, -60 millivolts. What would happen if a NA channel opened and stayed open? What about K? Na ions flow into cell until the membrane potential reaches 60 mV. K ions flow out of the cell until the membrane reaches minus 98 mV. What is the equilibrium potential? aka Nernst potential, reversal potential, membrane potential at equilibrium. How do we solve for equilibrium potential? Free energy Q, that represents electrical potential of membrane, delta GT equals RT lane, sin, Qt ZFE. Set equals to 0, 0 equals RT lane, sin, Qt ZFE, E equals RT lane, Qt, sin, ZFE, Na equals 60 mV, E, K equals minus 98 mV, F equals Faraday's constant, E equals equilibrium which ion actually moves against its concentration gradient when its gated ion channel is open in a neuron, Y. Chloride, driven by electrical gradient. Describe the events of depolarization of a neuronal membrane, a reaction potential. One, voltage-gated NOT channels to open for 1 millisecond, allowing depolarization wave to continue. 2, voltage gated K channels open, causing repolarization of membrane. 3, voltage-gated circa 2 near synaptic cleft open, causing localized increase in cytoplasmic circuit. 2, 4, fusion of vesicles at synaptic cleft, releasing neurotransmitter like acetylcholine. 5, acetylcholine binds receptors on adjacent neuron, allowing Na circuit 2 K ions through, causing AP to start on next neuron. What prevents an AP from moving backwards? Refractory period, when voltage-gated Na channels close and stay closed after opening. How does a voltage-gated Na channel operate? Voltage sensor, helix 4, normally pushed into membrane at resting potential, during depolarization. This helix moves out of the membrane, causing conformational change that opens channel, red loops. Form selectivity filter that only allows Na ions through at rate of 10^7 ions, sec. Channel stays open for millisec before green inactive ion gate domain binds and closes channel opening. Length of time channel stays open depends on length of green tether attaching gate to channel. How is the closing of Na voltage-gated channel thermodynamically controlled? Binding affinity of inactivation gate for channel opening. How is the closing of Na voltage-gated channel kinetically controlled? Length of tether, controlling entropy of system and number of unbound conformational states that this inactivation gate tether can occupy, screams entropy. What level of function is the action of the tethered gate on the Na voltage-gated channel? Mechanistic. What types of potent toxins known to exist in nature target ion channels? Snake venom, scorpion venom, venom from pufferfish, etc. Why are channels targeted by so many natural toxins? Binding an ion channel prevents thousands of ions from entering cell, or causes even more to enter. If gates stuck open, ions pass at rates near that of diffusion, big and fast to effect. Toxin doesn't have to cross the membrane to cause effect. What are integrins? Dimeric plasma membrane receptors bind RGD sequences like collagen, fibrinogen, and fibronectin. Also bind intracellular ligands like talin and vinculin to modulate assembly of actin-based cytoskeleton in the vicinity. What are the selective cell-cell interactions in which integrin plays a role? Embryonic development, blood clotting, immune cell fusions, tumor metastasis, and more. What two mechanisms are critical for integrins to bind extracellular ligands? Binding of divalent cations, e.g. circa 2, which brings ligand binding sites much closer to cell surface. Phosphorylation of extracellular domain, not well understood. How is integrin signal transduction accomplished? VMAP kinases like HERC signaling leads to changes in extracellular matrix by secretion of various proteins and factors. How do steroid receptors differ from other receptor signal transducers? Typically not in plasma membrane, along with gated ion channels are simplest transducers since they typically don't have a signal cascade. Describe the action of the steroid receptor. One, steroid hormone released from originating cell and binds a serum-binding protein. 2, hormone carried to target tissue and released to cross plasma, nuclear membrane to bind to receptor. 3, conformation of receptor then changes and binds other proteins to form hormone-receptor complexes. 4, complexes bind hormone response elements on DNA, altering transcription of specific genes. What are steroid hormones synthesized from? Isoprene, which are synthesized from acetyl units. How fast is hormone response to the activation of steroid receptors? Hours to days. Give 3 examples of steroid responses that are too fast to operate via transcription regulation. Estrogen-mediated dilation of blood vessels, several signal mechanisms involving GPCR-related signaling pathways, possibly one orphan GPCR binds estrogen. Which 4 signaling components are common to all mammals, plants, and bacteria? 1. Ion channels. 2. Electrogenic ion pumps. 3. Two-component his kinases. 4. Adenylyl cyclase. How do bacteria use the two-component his kinase signaling component? How old is the two-component his kinase system? Old. In prokaryotes and eukaryotes, chemotaxis is a cellular process used in most forms of life. Which two pathways highlight similarities in immune response between plants and animals? Plant pathogen defense pathways and animal receptors involved in innate immune responses. Describe the plant pathogen defense pathway. Receptor-like kinase binds to peptide from flagellin. Receptor activates MAPK cascade. Plant immune response elicited. Describe the pathway of animal receptors involved in innate immune responses. Animal toll-like receptors bind flagellin or lipopolysaccharide, activation of 2-MAPK cascades. Innate immune response. How old is the MAPK cascade? Found in eukaryotes, so as old as eukaryotes. Why don't bacteria have nuclear steroid receptors? Most bacteria don't have a nucleus. Steroids are hydrophobic molecules and need some sort of carrier protein. No need for bacteria to use this in place of so many other soluble and diffusible molecules available. Bacteria don't synthesize steroids. How is the signal transduction of electromagnetic radiation accomplished? In the light receptor rhodopsin in animals and bacteria. What is rhodopsin? GPCR with the chromophore 11Cis retinal linked by a Schiff base to the lysine of a protein called opsin, the photoreceptor seen in 10-9 rod cells in retina. What is retinal derived from, and what is that derived from, and eventually derived from? Vitamin A, from beta-carotene, eventually derived from isoprene units. What is the pathway of light to a neuronal signal? Light converts 11Cis retinal to all transretinal signal cascade through neuronal signal. What is the function of rhodopsin? Rhodopsin senses visible light near center of visible spectrum. What is the function of rod cells? High-resolution vision and night vision. What do different variants of opsin allow for? Where are these variants seen? Detection of light further in the red, green, and blue regions of the visible spectrum. 10-6 red, green, and blue cone cells in the retina. What is the function of cone cells? Color detection. Where is the detected light in rod and cone cells is interpreted? In several layers of interconnected neurons, which convey information to ganglion neurons of optic nerve. Biochemically, how do most forms of color blindness arise? Mutations in opsin, which is what binds to retinal. Where is rhodopsin in a rod cell? Describe signal transduction in vision. What entity is responsible for hyperpolarizing the plasma membrane during the AP? One photon of light activates, which activates a lot of, which closes, which changes the membrane potential by. One rhodopsin, 500 transducins, PDEs, 1000 ion channels, tilde 1 inhibits phosphorylation of the serine and 3N9 residues upon activation of rhodopsin in rod cells. Inhibited by SERCA2 binding protein recover and in the presence of high SERCA2. How long does it take for all transretinal to replace with 11-CI's retinal? What does this regenerate? Seconds to minutes. An active rhodopsin ready for the next photon of visible light. How is all transretinal converted back to 11-CI's retinal? Through a series of enzymatic reactions. Good enough. What is the purpose of the cellular regeneration of rhodopsin during signal transduction in vision? adaptation mechanism that can change the sensitivity of rod and cone cells by several orders of magnitude. What three proteins implement the adaptation mechanism of slow rhodopsin regeneration? Herastin 1, rhodopsin kinase, recoverin. What do light, smell, and taste reception have in common? One, start with GPCRs that have signal cascades ending with ion channels opening or closing. 2, many common components with other GPCR pathways, use of common second messengers. 3, self-inactivate. What is a hallmark feature of signal transduction pathways involving G proteins? Self-inactivation, even if help is needed to do so. What is the significant difference between sensory signal transduction pathways and other GPCR pathways? Phosphorylation, dephosphorylation event versus opening, closing of a channel. How does signal transduction pathways play a fundamental role in the regulation of the cell cycle? A family of heterodimeric proteins called CDKs control passage of cells through cell cycle. Regulation is through the cyclin subunit. How many CDKs and cyclins do animal cells have? 8 CDKs, 10 cyclins. In addition to regulatory subunit bindings, how is CDK activity regulated? Phosphorylation, dephosphorylation. Describe the regulation of CDK by phosphorylation, dephosphorylation. What specific events are required to activate a typical CDK? 1. Cyclin bindings. 2. Dephosphorylation of tyrosine. 3. Phosphorylation of threonine. Why are so many specific events required for the activation of a typical CDK? Huge commitment to start the cell cycle. Cell better be sure it's ready. Describe the timeline for significant cyclin-dependent kinase activity during the cell cycle. When does the cell become committed to mitosis and by what? When the cell passes the restriction point at end of G1 by activation of cyclin E-CDK2. What is the result of activation of cyclin E-CDK2 during mitosis? phosphorylation of PRB, which frees transcription factor E2F to promote transcription of DNA. Synthesis enzymes. When is p53 activated during mitosis instead of PRB? When DNA is damaged to the point of creating double-strand breaks. How does p53 function? Increases expression of p21, which inactivates cyclin ECDK2, preventing cell from passing the restriction point. What is the most important tumor suppressor gene? p53 mutations in p53 are seen in half of all cancers, often causing the crossover of benign to malignant growth. What is a tumor suppressor gene? A gene whose gene product inhibits a cell's progression to cancer, typically by restraining cell division or promoting apoptosis. How do cells die when they must? Apoptosis. When is apoptosis triggered? When cells are damaged, infected by viruses, or as part of embryonic development. What are two apoptotic signals? 1. Phase ligand. 2. Tumor necrosis factor. What is the structure of the receptors for the apoptotic signals? Membrane receptors that contain a cytoplasmic death domain that binds to the cytoplasmic proteins, which create the signals for cell death, a signal cascade leading to a family of cystine proteases, called capsases, and the DNA-cleaving enzyme adenylase. Describe the transduction pathway for apoptosis signals. Caspase 8 is an early initiator of the signal cascade leading to a family of cystine proteases, called capsases, and the DNA-cleaving enzyme adenylase. Caspase 8 proteolytically releases various proteins from the mitochondria, including cytochrome C. Cytochrome C activates many of the other caspases that then proteolyze most of the proteins in a cytoplasm. In what form do inactive caspases exist? Proenzyme. Many self-cleave to become active.